right, we're going to start now. I'll stop the music. And as always, there'll be some opportunities during this presentation for you students to ask questions. And so please use the chat window when we get there, and you get to interact with our speaker today. So our speaker today is Dr. Andrew Raub, LBHS Class of 2007, and I'm going to turn over the controls to him now.
So my background is in the area of organic chemistry, and most of my training has been in this area. And I'm currently working for a pharmaceutical company that is developing novel cancer therapies. So I'm going to tell you a little bit about how molecules are made, why they're made, and how we can use organic chemistry principles to translate biological discoveries into new therapies. So before I delve into that, I just want to go a little bit into my background. So my first foray into lab work was actually in ACR, where I worked on TAML catalysts, which we were using to oxidize carcinogens. And then after graduating from LBHS, I had an interest in chemistry in general, but I kind of wasn't really entirely sure what I wanted to do. I ended up actually starting out as an engineering major at Cal Poly SLO, and then moving on to community college afterwards to then kind of rediscover my interest in the physical sciences. And then ultimately at UCS San Diego, I was fortunate enough to work in two labs which kind of helped solidify my interest in chemistry. One was the Borvik lab, which we were primarily focused in inorganic chemistry, and then also the O'Connor lab, which was mostly involved in physical organic chemistry and organometallic chemistry. So after graduating from UCSD with my bachelor's, I worked for a short period of time at Novartis and got some relevant work experience in an area known as medicinal chemistry, which is where I currently work in. And then I also did a little bit of post-bac research in Switzerland. Then upon returning back to the States, I started my PhD at Stanford, where I worked initially in organic chemistry in the lab of Paul Wender, developing new synthetic methodologies and also new dye molecules. And then I worked in the lab of Daria Mochly-Rosen, investigating ligands that can actually promote protein-protein interactions. And now currently, like I had mentioned, I'm working as a medicinal chemist at Exelixis. So this is a brief overview of my talk. I'm going to talk to you a little bit about organic chemistry, how we make organic molecules and why we make organic molecules. Then I'm going to discuss a little bit about basic principles of drug action. And then I'm going to try to merge these two concepts and give you a bit of an example of how we would take a novel discovery in biology and translate that into a novel therapy. So organic chemistry, what is it? It's the study of the structure, function, and preparation of organic or carbon-containing organic molecules. Some applications of organic chemistry would include pharmaceuticals, petrochemicals, dyes, explosives, and cosmetics. And pharmaceuticals is what I'll be focusing on later. And an important concept in organic chemistry is that structure determines function of molecules. So I have three very different, in terms of structure, molecules here. The first compound on the left is a dihematine, and because of its arrangement of double bonds, it can absorb visible light, and this gives it the property of being a dye. Also, it has a unique shape which allows it to intercalate with DNA, and so it's commonly used as a stain for a nuclear stain in histology or visualizing tissues. Another molecule, TNT, which you're probably more familiar with, is an explosive, and what makes it such a potent explosive is actually, as you probably know, decomposition of organic matter or combustion with oxygen can be accelerated by the addition of extra equivalents of oxygen. So this molecule actually in it has additional equivalents of oxygen in these nitro groups. And so this basically allows it to not be as dependent on atmospheric oxygen and leads to a more rapid decomposition. And then another molecule pictured on the far right is penicillin. This is an important antibiotic that has saved an approximate number of 50 million to 100 million lives. And important basic feature of these molecules, so this is epanosine analogs, there are a variety of them, is this four-membered ring, with, which is called a lactam. And this chemical moiety is what allows it to inhibit the synthesis of bacterial cell walls, so they can't divide appropriately. So now that we know a little bit about why we want to make organic molecules, how is it done in practice? Well, this is actually a problem that I got when I first started my PhD, maybe the first month, where we were wanting to make this reagent here called the TME reagent. And it had never been made before, and we thought it might have some unique function. So we wanted to find a way to synthesize it in the lab. So commonly what you'll begin with in organic synthesis is a cheap, readily available starting material that you will then build in complexity through a series of organic transformations to your desired molecule. Now, what's involved in each of these transformations is an organic reaction. Organic reactions are comprised of three general steps, one being the synthesis of that molecule, which the bonds are being broken and formed in a reaction vessel like a flask. Then you have isolation, which allows you to selectively purify the desired product from this reaction mixture. And then characterization, to identify that you actually had made what you had wanted to make from the beginning. So this is kind of what that sequence would look like. So we have 4 different molecules here, the starting material, 2 intermediates, the desired product. And then between these molecules, we have an arrow, and above and below these arrows are just reaction conditions that, you know, when you subject the starting material to those conditions, you will transform it into the desired product. So there's 3 steps from commercially available starting materials to the desired product, but what is involved in one of these steps? So I'm just going to highlight this step here. We're taking this alkyne, we would say, and transforming it into these 2 double bonds. And first we gather our reagents, and you're probably familiar with this through some of your lab work at LBHS, and you have to measure the appropriate amounts, and then we would set them up in a reaction vessel. So pictured here, little flasks or vials on a stir plate, and there are some pink balloons, and we have pink balloons because we need ethylene gas to promote this reaction. And this is at room temperature, so there's no heating, just stirring. One important part of this is to monitor the reaction to make sure that you're producing the desired products. You can do that through mass spectrometry, which can look at the molecular weight of the things that you're making. Also, you can use things like TLC or chromatography to monitor the conversion of the starting material into the desired product. So one spot here would say is the alkyne in red, and the blue spot is the diene. So if that's the starting point of the reaction, and we're here, it looks like the reaction would be done. So now we have to purify away the undesired things and to leave only the desired product for the next transformation. So to do this, we use a variety of techniques. One would be chromatography, so a larger scale version of what we did here in monitoring the reaction. Another form of purification would be distillation, so taking advantage of differing boiling points of organic compounds. Extraction, which is taking advantage of different solubility properties in an immiscible mixture of, say, really non-polar solvent, organic solvent, and something very polar like water, or crystallization. And then finally, what we will do is we'll have to characterize the products from this reaction to make sure we had actually made what we'd hoped to make. And then we also can use things like NMR for this, or LCMS. And NMR is just a way to kind of look at the nuclei or the atoms in space, so it gives us an idea of the connectivity and the structure of the molecule we made. And LCMS is similar to mass spectrometry in that we can get an idea of the molecular formula that we have made. And often when you do this, you know, things might not come out as pure as you'd want them for the application they're using. So we'll kind of resubject them to the purification process, and then also characterization process again. So you can see this kind of a labor-intensive process. To go from step 1 to step 5, it could take, you know, let's say an hour, average of 24 hours per reaction. And because it's such a labor-intensive process, one goal in organic chemistry is to basically develop new reactions that can shorten the time frame that it takes to make molecules. Fortunately for us, I said the TME reagent would have some unique function, and that function is being able to make a whole bunch of bonds in a single reaction. So if you compare the complexity that we generated in this step, where we added basically 2 new carbon fragments, we can take this TME reagent and stitch together 2 other molecules in a single step. So for our 24-hour reaction period, we can generate a whole bunch of complexity, which shortens the time it takes to make useful things. And then subjecting this to a couple more reactions. One is an oxidation, we call oxidation. Another, we would put in this amine group. We can take a material that is fluorescent, but it can't sense its microenvironment. So these are a variety of different solvents of different polarity, and you can see that the molecule fluoresces blue in all of them. And by installing this nitrogen, we can create a new class of dyes that actually can sense its microenvironment. So we have things like DMSO, which is a very polar water miscible organic solvent, and then going all the way to cyclohexane, which you could think of as like a really non-polar, something that would be like gasoline, right? And so this molecule actually has a property that it can change its emission spectrum based on the environment, which is very useful for things like monitoring binding events or protein-protein interactions or conformational changes. So we were able to basically kind of create a new class of dyes through creative reaction science. So as a summary for the first part of my talk, an important part of organic chemistry or concept in organic chemistry is that molecular structure determines function. And the example I used for that was, one of them was TNT, where it carries equivalents of oxidant to lead to a really aggressive chemical decomposition or combustion. Organic molecules are synthesized in a stepwise fashion from less complex starting material. So we had this alkyne, which we used through a variety of steps to make this useful reagent, TME. And then with an understanding of how molecules react and then how they behave, we can then use a creative reagent to then create a new class of dyes in a really expedient and efficient manner. So I think that is where I'll take questions for my first section.
All right, so students, please put your questions in the chat, and the teachers will be monitoring that. I'm going to ask a question while I'm waiting for students to formulate their chat questions, but other teachers, you can also feel free to ask questions. So my question is this: you made it look so pretty, and you just do step 1 and step 2 and step 3, and then you're done. And I'm wondering, how long did it take you to figure out how to do step 1 and step 2 and step 3?
Okay. Yeah, that's a great question. So Steve's wondering, is this something that worked exactly as you expected on paper? Was there any other process to developing this reaction scheme? And you're right. Actually, I was going to put this in here, but the first attempt at making this TME molecule, we started from a different precursor using different reactions. And we got halfway through that synthesis. So sometimes, you'll come up with a variety of different routes. You'll start with one maybe that is the fastest or most easy to quickly investigate. And then, sometimes you'll be met with failure after a couple of attempts, and then you'll instead switch to a different reaction scheme. And so this one ended up probably taking about a month to make. So the first few different iterations or first iteration wasn't successful, but this one was readily accessible.
Excellent. Okay. Here's a question from Luca Elganian. What is TMS?
Trimethylsilane. So it's basically a silicon atom bound with 3 different methyl groups. And it has an important function in this molecule in that it's a source of electrons. So it allows for the unique reactivity that we see in this molecule in this step. So it's an important feature of this compound.
Yeah, you've got a lot of abbreviations there, don't you?
Yeah, sorry. I don't want to go too deep into it.
Alphabet soup. All right. Here's a question from Bronson Bailey's. It's an interesting question to ponder. Could you use organic chemistry to fix Down syndrome when you have a Down's infant?
Yeah, I think that, so I'm not too familiar with targets that you'd want to try to make molecules for in Down syndrome, But certainly, I think for a lot of CNS diseases, there are molecular targets that we can investigate to kind of ameliorate certain, symptoms. So like things like Parkinson's or other types of CNS diseases, there are targets that we can exploit. But I'm just not entirely sure about Down syndrome itself.
Once again, you used your alphabet soup. CNS stands for?
Oh, central nervous system, sorry.
Yep.
So targets
so
kind of in the brain.
Yeah, the jargon where you just say CNS. Yeah, I mean, all your colleagues know what you mean. Yeah. Okay. So
Yeah.
nervous system disorders. You mentioned Parkinson's and depression, probably, huh? Yeah.
Yeah, that's an.
Okay, great. I think we'll go ahead and let you go on with the rest of your talk now. Well, okay. This one, this is going to be a real challenge for you.
Okay.
Why and how would you use NMR?
So NMR is very useful because it can tell you how molecules are bonded together. So it can tell you basically the spatial arrangement of them. So you could have something that's called an isomer, which has the same number of molecules. So in mass spec, it'd have the same formula. But the thing is, things can have the same formula, but different structure. And that's when NMR is very useful. And how do you use it? It's another form of, it basically puts molecules in a permanent magnetic field. And depending on how much electron density they have, how many electrons, you can kind of get some energy from, this is gonna be too hard. I would need a slide for this.
But there's a picture of the NMR there, which we don't get a sense of scale. It looks like it's about the size of a Kleenex box, but really that's pretty big, isn't it?
Yeah.
Yeah. So is that human sized, or is there a reflection of humans in it? Maybe. Cool.
It's probably 6 feet tall, 7 feet tall.
Okay, so it's a big machine, and it costs a lot of money, and very powerful tool. All right, we'll let you go on now with your talk.
All right. All right, so the second part of my talk is going to be discussing some basic principles in drug action. So first, probably to start with the definition of a drug, which is a substance that when introduced in the body produces a measurable physiological effect. And certain examples of physiological effects could include lowering blood pressure, sleep induction, alterations in blood glucose levels. And drugs can broadly be classified into two categories. One is our small molecule drugs, and the second are biological drugs. So on the left, we have some examples of small molecules. These can range in size from 18 atoms, like something that's used in multiple sclerosis, up to 113 atoms. This is Taxol, which is actually used as an anti-cancer agent. And what makes small molecules different from biological molecules are, usually they are single molecular entities that are easily characterized. They're made via chemical synthesis, kind of in a manner akin to what I'd shown you in the previous section. They're lower molecular weight than biological molecules. And so this kind of confers them some unique properties that kind of complement biologics. And they can be administered using a variety of routes. So they can be administered orally or intravenously or intranasally, or a variety of different ways. And biologics, in contrast, are kind of difficult to characterize because of heterogeneity. So these molecules are made of, I guess we could say, amino acids and different structures. So insulin's a small one, 784 atoms we can have, and it's a hormone. We can have something like an enzyme, which is much larger, like tens of thousands of atoms. Or we could have something much larger than that, which would be a humanized antibody. And these are made through protein expression in mammalian cells typically. And they have very high molecular weights, and they have to be administered via IV because they're peptides. They usually are degraded upon oral administration by the acid in your gut. So to give you an idea of kind of the relative number of molecules, small molecules versus biologics, in 2019, there were 38 new small molecules and 10 biological drugs approved by the FDA. So while small molecules probably have a larger share of the drug market, biologics are also a really important class of molecules that are kind of increasing in number and scope. So since my training is in organic chemistry and I'm a medicinal chemist, I'm going to be talking about small molecule drugs. So I said that small molecules have kind of unique properties, and one important property is that they can readily diffuse through cell membranes because they're both soluble in water to some degree and organic solvents. And you can think of a lipid membrane in a cell kind of like an organic solvent. It's very nonpolar. So cells are composed of mostly water, but other things are composed of largely of proteins, and these are actually the drug targets that molecules enact on. And so having a drug that can, and proteins are obviously located throughout the cell. So having a molecule which can diffuse through the cell membrane and say associate with proteins on the nuclear envelope confers it a wider range of applications sometimes than say a biological drug, which can only say associate with the surface of the cell. So here are some examples of drugs and drug targets. So the human genome is about 20,000 genes and functional genes, and about 1% of these are targeted by approved drugs. So one large class of drug targets are GPCRs, and these are often targets found in the central nervous system, CNS, and one molecule, atenolol, can actually treat high blood pressure by blocking beta-adrenergic receptors. These are basically like receptors that recognize adrenaline. And another class of compounds would be ion channel modulators. So one example of this would be propofol, which actually induces anesthesia or loss of consciousness, and this is very important for use in surgery. The history of surgery before anesthesia is quite gruesome, and this does it by modulating GABA. Another class of protein targets are nuclear receptors, and so these would be targets like recognizing hormones that change gene expression, and one compound representative of this class is tamoxifen, which is used to treat breast cancer as a selective estrogen receptor modulator. And then other could be a variety of enzyme targets in other pathways. And this one is, you can imagine it's being used in lipid synthesis or cholesterol synthesis, and it's actually an inhibitor of that, so it can treat high cholesterol. Another important small slice of that pie chart are kinases, which I'll touch on later. And these are very important anti-cancer targets. So how do drugs work? Drugs work by binding to receptors, and upon binding, they alter their function. And so a simple example of this is, I'll show you an enzyme inhibitor. So enzymes recognize substrates, they catalyze a chemical reaction that leads to the formation of products or other small molecules. And here's a little diagram where you have the enzyme and an active site where this chemical transformation takes place. You'll have the processing into the substrate into products, and then those will leave, and that will be one catalytic kind of cycle of the enzyme. And a competitive blocker or small molecule inhibitor inhibits the ability of the enzyme to process its normal substrates. So you have the active site, and the competitive blocker actually goes in and fills up that active site, which prevents the substrate from coming in and performing its reaction. But that's kind of an overview of how inhibitors work, but how do they actually bind to receptors? So two important concepts in this area would be shape complementarity. So a nice analogy would be you could imagine a receptor's lock, and small molecules are a key, and there's a specific spatial arrangement of their features that allows for one key to fit while another can't, and obviously to turn the mechanism. So similarly to a lock and key, receptors, active sites have specific geometry and volume, and small molecules which complement that shape can fit into them. But if you try to build a chemical group that's too large to fit in that space, there's just no way that that's going to bind. So you have to make molecules that kind of obey the space that is available to you. So another concept would be in this area would be electrostatic interactions. And proteins and drugs have areas of positive charge and negative charge. And ideally, when you're making molecules, you want to make sure that the areas of positive charge interact with the negative charge, leading to some sort of productive plus minus opposites attract interaction. If you build a drug into a receptor site that has a positive charge, and you also place a positive charge on your drug, that's going to be a repulsive force, and so you're never going to get it in there. So you kind of have to balance size requirements with electrostatic requirements when making these drugs. So this is a brief summary of what I just talked about in drug action. So drug is a substance that, when introduced into the body, produces a measurable physiological effect. And these drugs work by binding to their receptors and altering their function. And you can imagine drugs and receptors kind of behave like a lock and a key. So the keys being the molecules and the locks being the receptors. And electrostatic forces between ligands and drugs are important for the associations of these two different molecules. And this is another area where I'll take some questions.
Okay, so once again, students, please use the chat window to pose some questions for Dr. Raub. And once again, I have a question for you. So if you go back, I think it's just one slide. Yeah, that one there. Okay, so in the colorful picture, not the cat, but the colorful picture, the thing in the middle looks like a person, but presumably that's the drug there?
Yeah, that's correct. So this is a kinase inhibitor. So this is a drug molecule.
Okay.
And that's the gray area is kind of the volume that drug occupies. And then the green, red, and blue area is the surface of the receptor. So you can see kind of neatly fits in that area. So
the green, blue, red, that's the protein.
That's the protein, yes.
The enzyme. That's the enzyme.
Yeah.
And you're trying to fill the slot, just like you had in the previous slide, where you're saying if you put something in the slot, then the enzyme won't be able to do its thing, huh?
Mm-hmm.
Okay.
Yeah, that's correct.
And what are the colors, the blue and red and green?
So I guess this is kind of what is important for considering in terms of electrostatic interactions. So green is really kind of non-polar. So this is something that's organic-like, whereas the red and the green are kind of associations between red is negatively charged, green is positively charged. So we could potentially take advantage of these areas of negative and positive charge to kind of build in kind of elements in our drug that would complement them.
Okay, and the drug is much smaller than the protein, huh?
Yeah, so...
what do you mean by small molecule, huh?
Yeah,
okay.
A few hundredfold smaller.
Yes, okay, all right, we've got some questions now I can relay to you. So Anna Chang asks, what is a ligand?
Oh, ligand, so sorry. Ligand would be drug molecule. Ligand is a synonym for a drug. And we could use them interchangeably, so.
Okay, so in this picture, the thing that looks like a Lego person, that's the ligand.
Yeah, that is also the ligand.
Binding to the protein. Okay, good. All right, second question is asking about the cat. And the question itself is the cat okay? But you could probably talk more about what is that picture there for.
I think the cat's upset, but I think it's gonna be fine.
So how did that picture come to be?
I don't know, some random Googling.
Okay.
It's not my cat, so.
It's covered with styrofoam, isn't it?
Yeah, I'm assuming cats love boxes, so I think it probably jumped in and created its own field of electrostatic interactions.
And the styrofoam is sticking to it. Some of you may, if you were in my class, Bastian Evans was really fascinated by this, where I rubbed the styrofoam cup with the paper towel and then it would stick to my hand. And so that's probably what's happening with that cat.
Yeah.
That it's the styrofoam is sticking through the attractions of the plus and the minuses, which we call static electricity. Okay, I think we'll let you go on with your talk.
Okay. All right, so for the final portion of this, excuse me. All right, for the final portion of this, I'm going to kind of merge the two concepts that I highlighted in the first sections of organic chemistry and drug action to then give you an example of how we might use these concepts to make a drug for some novel biology. So the indication that we're going to be looking at is cancer. Cancer is characterized by rapid cell division and also by infiltration into other tissues. So this uncontrolled cell growth leads to the formation of tumors, and these tumors kind of interfere with function of local tissues and organs. And healthy cells transform into cancerous cells through genetic lesions or mutations, and these mutations can result from either some environmental factors, for example, UV irradiation, like in skin cancer, or nicotine in terms of lung cancer. And usually these mutations will, I guess, modify the cell's functioning in areas of metabolism, differentiation, proliferation, and migration, and that those functions allow them to grow rapidly and also to leave the site where they first mutate and form. And I kind of mentioned that kinases are an important class of anti-cancer targets, and one kinase that's an important anti-cancer target is c-Met. And kinases are enzymes, is like kind of the example I'd shown you with enzyme inhibitor, in that they basically cause the transfer of a phosphate from ATP onto a substrate molecule. And c-Met is a receptor tyrosine kinase. So, and it's also, so it's found at the cell surface. So this is the lipid membrane here. We have our c-Met receptor. And when its ligand binds, which is HGF, it causes a signal transduction cascade through these pathways.
What's HGF?
Hepatocyte growth factor.
So it's a growth factor.
Mm-hmm.
And so this I think is going to lead back to Lisa Situ's talk, where the cancer cells getting an aberrant or an overactive growth signal.
Mm-hmm. Yeah, that's correct.
Good.
Yeah, so mutations in c-Met, or I guess overexpression of the growth factor, can lead to overactivation and signaling through these pathways and lead to, I guess, what would be a phenotype associated with the disease state of cancer. So cells that won't die when they're supposed to, cells that proliferate when they're supposed to stop, and also that are souped up because of alterations in metabolism. And c-Met is highly correlated with poor prognosis. So overactivation, this can be overexpression, so a larger number of c-Met receptors, or heightened c-Met activity is correlated with poor prognosis in kidney cancer and liver cancer. And so, how do we use this information to then slow the progression of cancers that are implicated with c-Met? So, kind of like our example in the second part of the talk, we would use or develop a small molecule inhibitor of c-Met to then try to intervene and slow down the progression of cancer. So where do we start? Usually a target-based drug discovery. So our target is c-Met. We'll begin with a high-throughput screen. And before we can start this high-throughput screen, we'll develop a reliable assay to measure the activity of our c-Met in its enzymatic function. So this is transferring a phosphate from ATP onto a substrate molecule. And once we have a reliable assay developed, we'll then screen our collection of molecules against these targets. So in the case of Exelixis, where I work, we have about 5 million compound library. And so to screen such a large number of compounds, and this is kind of a picture of what a compound repository would look like, we have to use robotics to iteratively screen. And then once you've screened the collection of molecules, you'll see that some molecules appear to be able to inhibit the enzyme function. So c-Met would be less active, less good at what it's doing, relative to control or other molecules which have no affinity for the receptor. And so we'll validate the results and then identify a chemical starting point to start making a drug.
So how long does that screen take? It's 4 million compounds.
So you could, I think, so I'm involved in the medicinal chemistry side, and I will give you an approximation of how long that takes.
Okay.
I believe that you can run a high throughput screen in the order of a business quarter or less, so a couple months, 2 months maybe. I think, once you have the assay that's developed and you're comfortable with how sensitive it is, then it can be done, in a few weeks. because these things, robots can be working around the clock,
Okay.
screening many, many, many compounds.
But it's at least a few weeks to screen all those compounds, and then
Yeah.
hoping to get something that's active. And okay, good. Thank you.
Yeah. And so from our high throughput screen, we've identified one molecule which is active, but it doesn't have the necessary properties that we would want for a clinical candidate. So I guess in short, it can inhibit c-Met, but maybe it's not soluble enough. Maybe we have some concerns from toxicity perspective. It's not maybe absorbed well into in vivo systems, so like into the bloodstream of the mouse, for example. So this is where my work in the organization is focused. So taking this initial hit molecule and then using organic chemistry principles to then evolve it into what would be a clinical candidate or a drug. So where we start with this is making synthetically accessible modifications to the molecule. So using kind of principles outlined in the first section where we're developing that TME reagent, but identify ways we can construct this molecule that will lead to rapid synthesis. Because we only have maybe a year or a few years to run this project. So we can't, be doing things that are too difficult or take too much time. In addition to making transformations that are fast, we also have to make transformations that make sense. There's probably an infinite or nearly infinite number of compounds in this chemical space that you could be making or that are drug-like, but obviously only a limited number of resources and time. So we want to basically make molecules that will kind of fit the criteria that I set out in the second part, which was in the locking key and electrostatic example. So fortunately, shortly after our high throughput screen, we were able to get an X-ray crystal structure of one of our proteins in complex or c-Met complex with our drug molecule or ligand. And so this is basically showing you the shape of the receptor with the molecule crystallized or docked into that site. And so we can use that to kind of make sure we build in portions of the molecule that aren't going to be too big to fit into the receptor. And in addition to I guess this concept, we'll be building in portions of the molecule that can make favorable electrostatic interactions. So this was kind of similar to that diagram with the red, green, and blue surface of the receptor. These are just, in the gray, are different residues or amino acids on the protein, and we will then build in the molecule such that there's positive interactions with those residues. And so what that would look like kind of is we would then be breaking down this molecule into portions that we could easily make and that also make sense, that are rational. So then we'll transform this group maybe into this aromatic ring, or we'll transform parts of this linker. And not only are we trying to optimize the ability of the drug to bind and inhibit the enzyme, we're also trying to optimize things like solubility and permeability. And so those are really important features of what would constitute a good drug. So now once we have a molecule that we feel relatively confident about in its ability to inhibit the enzyme and also its solubility and other properties, we'll then move it into higher model studies. So this would be efficacy models or toxicity models. So a common efficacy model in cancer is basically involves implanting a humanized tumor, so human cells, into a mouse and growing them to a certain volume. And then you either start with a drug treatment once they reach a certain volume, and obviously you have to have your control, which is pictured in black. So we advanced two compounds from our medicinal chemistry efforts, this 2883 and 1820, into this efficacy model to see if it can in fact shrink tumors and validate our hypothesis targeting c-Met. And excitingly, we can actually see that relative to the control, in which the tumor rapidly increases in size over the course of a month. Treatment at about 10 mg per kg of our two compounds kind of causes the tumor to go into stasis. So it's not expanding in volume readily. So there's some positive results, but one negative result is that the compound in red actually has some sort of toxicity issues. And that's why there's only a couple data points. So we found out that, okay, at a higher dose, we have efficacy, but now we have toxicity. And this gives you the concept of therapeutic window. So you want to basically make sure that the toxic effects that you observe of the drug are actually orders of magnitude, ideally higher than the dose that you need for efficacy, i.e. reducing tumor volume or size. So, we'll have some we'll have some positive results in the sense of efficacy, but we'll need to go back and work on the toxicity of our molecules. So this kind of feeds back into the general workflow of medicinal chemistry, where we're synthesizing compounds using, our stepwise chemical synthesis, subjecting them to easy, fast in vitro assays, and then we're putting those promising compounds back into in vivo studies, toxicology, efficacy, and then formulating new hypotheses on how we can improve upon those drugs given the assay results, and then subjecting them back into the cascade of assays. So once we've gone through this enough times to feel like we have a compound that has a great chance of success in the clinic, we'll then do what are IND or investigational new drug studies. And these are basically very rigorous studies looking at safety profiles and efficacy of the compound to then present to the FDA. And then once the FDA looks at those IND studies and says, yeah, it looks safe, and it looks like we'll have a reasonable expectation of benefit for patients, you can then initiate a clinical trial. And these are done in phases 1, 2, and 3 generally, with phase 1 being looked at mostly for safety and tolerability, and then the expanding phases of 2 and 3 look more at efficacy. And pending positive results from the clinical trials, you get a new approved FDA-approved drug. So, this example I've shown you, this is a result of this was done, or a version of this was done by Exelixis, and the compound that ultimately resulted from these medicinal chemistry efforts, drug discovery efforts, was cabozantinib. And this project, which was targeting c-Met, started in 2002 in the discovery phase. So basically the medicinal chemistry screening efforts was completed in 2 years. And about 5,000 compounds were registered, to give you an idea of the time it takes and the number of steps are involved in this process. And then after about additional 10 years from finishing what you call the preclinical stage or the research stage, to then an approved drug was 10 years. So this whole process from beginning to end took 12 years. And ultimately, this compound was approved for use in thyroid cancer, and then kidney cancer and liver cancer, and thus validating our idea of targeting c-Met in the first place for these kinds of tumors. So in summary, we started with a disease indication or an unmet medical need, which was cancer. We identified a novel target, which is implicated in really aggressive forms of cancer. And we then decided to basically make an inhibitor of that enzyme. And to do that, we had to create an enzyme assay looking at how active our enzyme is, and then treat all of our compounds in our compound library in this kinase assay. And ultimately, we got a one-hit molecule, which was able to inhibit c-Met, but it wasn't able to, get into the bloodstream, or it didn't have solubility that we were happy with, and a variety of other properties. And through rational drug design and organic chemistry, we're able to then arrive at a drug like cabozantinib, which is suitable for clinical trials. And ultimately, after additional 10 years in clinical development, we can arrive at a compound that is finally FDA approved. And I think, yeah, that's the last slide, and I'd be happy to take questions on this or any other parts of the talk.
Okay, once again, students, try to put a question in the chat. So I want you to try to talk about the example you talked about here is not what you're currently working on. Mm-hmm. So what is the big need for the secrecy?
Well, okay, so part of it, you couldn't really share any chemical structures that are being made. And that's partly because of the things that we're working on now, we don't actually have IP on yet. So their patents are being filed or have to be filed, right? And if you have a public disclosure of a structure before you file a patent or before this patent's approved, it can basically invalidate any of the novel things that you're working on.
Interesting.
So,
Okay, you're trying to get a patent, and how encompassing is a patent? If you're saying, is it just this particular structure, or is it
yeah,
part of the things that are like this as well?
patent. It's a bit of both. So you can get what is called a composition of matter patent. So this would basically be a single molecular entity. Like we could patent this molecule and have exclusive rights to however that molecule is used because it had never been made before. But in the process of going from here to here, we can also define a whole bunch of molecules, which will basically prevent, I guess, kind of other companies from developing similar molecules, or what is called patent busting. So basically making a small modification, so like just removing a fluorine or something, because that's a way to, I guess, kind of protect your novel contribution to this area. So it's both getting a single molecule or a series of molecules patented that you have exclusive rights to, but also kind of patenting an area of molecules. It's a bit of both.
Yeah, so it's very interesting. Presumably, with a target like that CMET that I'm looking at right now, there would be other drug companies saying that's a target, we should make something to inhibit it.
Yeah, that's correct. And there are other compounds that have activity against CMET. And I guess one thing that I didn't go into too much detail in this or even touch upon is that there are hundreds of kinases, and we're basically inhibiting the active site. So the active site it's kind of mimicking ATP. So compounds actually have a variety of potencies against different targets. So CMET is one of the targets. There could be other kinases that are targeted. So typically companies will, I guess, put a stake in the ground based on what they get out of their proprietary chemical screen or their high throughput screen. And so while other company might target CMET, it's probably going to target a whole bunch of other things.
Okay.
And that the profile of its kinase selectivity will kind of confer it certain advantages. So yes, companies are kind of in the same space, but they kind of differentiate themselves based on the unique structures that they pursue.
Okay, so a different company might come up with an inhibitor that looks very different from what you have there, huh?
And it might benefit from some of the pharmacology of the ligand that we have here, the drug that we have here, but it'll probably be distinct in a variety of other ways.
Yeah, okay. Okay, here's a question from Grayson Joseph. Let's see if I can read it. All right, what happens if the active site of CMET mutates to such an extent that the molecular geometry is too different for a drug like this carbametics or cabozantinib to properly inhibit?
Yeah, that's a great point. So that can happen, and that does happen, and that's just a general way that cancers can mutate and confer resistance to certain chemotherapies. And typically what is done, CMET is one target for, say, like kidney cancer, but there are other targets that are implicated in the progression of that disease. So sometimes what people will do will either add more than one target. So they'll add maybe an antibody and your ligand or your drug that targets CMET. So if one pathway gets mutated, there will still be anti-cancer activity from another drug. Or you will say that, okay, CMET's now mutated, so now we're going to put in a different kinase inhibitor. So we'll then switch to a totally different target, because now the patient isn't getting the benefit that they were getting originally from this compound, and it has, certain toxicities associated with it. So if you're not going to get any benefit from it, then you don't want to use that chemotherapy. And there are other ways of going around that, but that might get a bit involved.
Yeah, so I'm trying to think about that. So to make this one drug, you're saying took many years. Part of it in the laboratory making the different molecules. Did you say it was 5,000 different molecules were made before it was decided this one's the best?
Yeah.
Yeah. So you have to make all the molecules and decide which one you're going to submit to FDA trials.
Mm-hmm.
And so there's all that time developing it, and then there's all the time doing the trials, which you say can also take years. And so it's hard to see how, I'm almost saying it's hard to see how do you make a profit out of that? It's so long to develop, and presumably you have a limited number of patients who can benefit from this new drug.
Mm-hmm.
And so you've got to make enough on those patients to justify all the investment of time and manpower and machine power to make it so that, yeah, we're still a company that makes money.
Yeah. I'm kind of being more on the science side. I don't know as much about the business development side, but I mean, before they initiate a drug discovery effort, they certainly look kind of at, what could the impact be of a successful outcome, right? So how many patients have this disease? What type of benefit do we expect to add to their lives?
Mm-hmm.
And that gives you an idea of basically how much you can charge for the drug. And there are healthcare systems that, require a certain amount of, quality of life improvement at a certain price tag, basically. So you can't charge above that. And I can't really say too much more beyond that because that's not really my area. Yeah. But I think that people look before, well before they start these projects to make sure that they can recoup their investment.
Yeah, so you wouldn't start the project if you didn't say, we have a model that shows we'll make money on this if we're successful. Okay.
Yeah, and that's something that doesn't entirely sit well with me.
Oh yeah, it seems really bad to say, well, we're not going to help those people. They don't make enough money. We can't make enough money off of them, so we won't develop the drugs that can help them.
Usually the way that you could spread the risk of the cost of pharmaceutical companies recouping the investment is obviously through, larger insurance system. So, and also having the ability to bargain against pharmaceutical companies for a lower drug price is really important.
Mm-hmm. Okay, I had one more question. But I forgot what it was. Can I get it back? Maybe not. All right, it's noon. So I think we'll say that is the end of this webinar. Thank you all for being here. And there will be one more ACR alumni webinar next week. It'll be on a completely different topic. It'll be on archaeology. But it'll be another example of science in the real world. So I will close this meeting for now. So time to say your goodbyes.
All right.
Thank you.
Okay, we're getting lots and lots of thank yous in the chat and a few out loud. All right, so why don't we, what we'll do now, Andrew, is we'll have a little debrief from the teachers who are here. And there's from Grayson with a very personal one. Thank you, good luck with your drug in regards to the FDA.
Thank you.
Okay, Cameron T says, great presentation, thank you. Most people are just saying thank you, thank you. Okay, good. All right, so co-hosts, let's go ahead and get rid of the people who don't get rid of themselves. Why don't we do it this way, huh? All right, it's a little tedious. We have to click and say remove person. And do you really want to remove? Yes. So 3 clicks to get rid of somebody. All right, but we have 3 teachers who can do that. And They'll be gone soon enough. Click, click, click. Now we're down to single digits. 3, 2, 1. That should be it. Okay, good. Oh, Nick Brown is here. Do you know Nick Brown?
Yeah.
Andrew? You do? Okay.
Yeah.
He's joining us. Yay. He's doing next week's webinar. So I'll get a chance to talk to him. All right. Well, let's do this without, well, let's let the teachers decide. Do you want to do this with the screen share so we can ask about specific things? Or would you like Andrew to stop his screen share so we just see the humans?
I like seeing the screen.
Okay. All right. Why is Abby still here?
I thought I kicked her out.
Yeah.
Yeah, it won't do anything when I click it.
I mean, she's gone. She's just. So yesterday I found there were a lot of people I could not assign to breakout rooms, so I had to send them back to the waiting room and then readmit them, and then they showed up on my list of people I could assign. So that was good for me to learn. A little late in the year, but good for me to learn. All right. Nick Brown is a pirate. Are you there, Nick?
Hi, everyone.
Hello. There you are.
You
Okay.
guys.
Wonderful. Wonderful. Good to see you.
Great talk, Andrew.
Oh, thank you.
All right. So give some feedback to Andrew about the presentation there, or ask some questions.
That was extremely interesting and very complex.
Thanks. Yeah. Thanks for keeping me honest with the jargon. I haven't actually had to give a talk to high school students probably since the last time I talked to ACR back in 2013. So this has been a very good experience for me.
Yeah, it's easy to fall into the jargon, and you forget that most of like your audience. But yeah, that was really informative, and I didn't realize bringing a drug to market was so laborious and complex and time consuming.
Yeah, it certainly is.
I would imagine that the business side of things is they have drugs that they know they make a lot of money off of, and then they can fund the like smaller, more drugs like this that are less widespread, I should say.
Yeah, I see. I think that companies kind of go after areas of their expertise to some degree. So they're not all going after what's going to be the next blockbuster entirely. So my company is kind of in the area of oncology. So we'll kind of pursue targets that are probably like, can have a large market impact, but also that maybe have a smaller one. But we have a higher degree of confidence that we'll be successful based on our expertise. So
got it.
Yeah, so I think to some degree there's that going on. But yeah, not everything's going to be the next Lipitor.
Understandable.
So how much turnover is there at your company?
In terms of scientists?
Yeah.
I'm trying to think how many. So in the chemistry department, we've only had like one intern, and that was before I started leave. So low turnover in the chemistry department, which is a good sign.
Yeah.
The biology department is actually much larger than the chemistry department. We only have about 15 chemists in total. But in the biology side, so this would include high throughput screening. This would include, people doing cellular biology. That side of the company is about 60 to 80 people.
Wow.
And turnover, I think since I've joined, we had one person leave. So pretty low as well.
Yeah, it's really good.
Yeah. I've seen smaller labs in grad school with like 10 people have higher turnover than the company.
So if you go to another company, then you know some secrets. And so how is that dealt with?
Yeah, that's a good question. I've actually wondered that myself. And I think it's kind of talked about sort of like hush hush amongst the professionals in the area. But since I haven't really had another employment outside of Exelexis, I wouldn't really know how that is in practice. I think that sometimes there are companies that kind of hire people with expertise in a specific area. But obviously, you're not gonna, as a chemist, you're not gonna jump into the same chemical space that we were just working on at another company if that were to happen.
I would imagine that there's some sort of contract language that says there's a certain amount of years where you can't work in the same area.
Oh, like a non-compete?
Yeah, like a non-compete maybe, where it's like you can go to a different chemical company, but you can't make cancer drugs.
Yeah, that's an interesting idea. There may be. I'll probably have to ask my advisor or my supervisor if that's something that happens. When I had an internship at Novartis for a bit, so I got to see a lot of the things that they were working on, and I didn't have to sign anything. They kind of like, I don't know, they didn't really give me any debrief on what I can and cannot talk about. I just understood that I can't show anyone any structures.
Interesting.
Yeah.
Well, I guess if there's really low turnover, then it must be a wonderful work environment for you.
Yeah, it's been really great. Yeah, there's
I'm sorry, I don't know if I missed this.
Oh, sorry.
Where's the lab located?
It's pretty close to Oakland.
Oh, okay.
So on the East Bay. Yeah, it's very different to work for a large research organization that has a few projects and a lot of people as opposed to academia. You know, it's like a big change. But I think for me, I kind of like being on a large team and having it be so multidisciplinary.